are we ready to go? I know.
Yeah, we're ready.
All right. In 3, 2.
Do not engage an enemy more powerful than you. These are the words of famous military tactician Sun Tzu, who wrote The Art of War. According to this logic, if you know that the enemy is more powerful and therefore able to defeat you, then you shouldn't engage them in the first place. Cancer seems like the all-powerful, undefeatable enemy. And according to this logic, we shouldn't even try to go up against it. However, this is a choice that we as humanity do not have. We must continue to challenge cancer, no matter how impossible the odds may seem. And why? Because of a disease of this magnitude that has had such a devastating impact on so many people's lives, the only option we have is to engage the enemy. Today we will be talking about soft tissue sarcomas, a rare type of cancer, and how Trabectedin, our drug, has an impact on it.
So, what is soft tissue sarcoma? Soft tissue sarcoma is a rare type of cancer that develops in the soft tissue of the body. It can develop basically anywhere in the body, but there are specific types of soft tissue sarcoma. Our drug, Trabectedin, treats liposarcoma and leiomyosarcoma. Liposarcoma is a cancer in the fat tissue. It is most commonly found in the thigh area. Leiomyosarcoma is a cancer in the smooth muscle tissue. It is most commonly found in places like the uterus or the small intestine. To show the rarity of soft tissue sarcoma, we're going to show how the diagnosis compares to lung cancer, the most commonly diagnosed cancer in the United States. While soft tissue sarcoma is diagnosed 18 times less than lung cancer in 2015. So, what are the other soft tissue sarcoma therapies? So, another soft tissue sarcoma therapy is Trabectedin. It is a chemotherapy administered by an IV. And it treats patients that have melanoma, a type of soft tissue sarcoma, and it's a skin cancer. And it's given to patients where the melanoma has spread to different parts of the body away from the tumor. So that means their melanoma has advanced outside of the initial stages where the cancer has moved away from the tumor. So the mechanism of Trabectedin is it methylates guanine at the N7O6 positions, and the DNA strands stick together, which will then lead the cell into apoptosis because the cell cannot replicate. And Trabectedin affects both healthy and cancerous cells. So, background on Trabectedin. It was developed under the brand name Yondelis, and it was recently approved on October 23rd of 2015. The method of administration is by an IV, and it was isolated by the marine organism, the mangrove tunicate, which is seen right here. And it was approved for the treatment of the soft tissue sarcomas, liposarcoma, and leiomyosarcoma that is more advanced and cannot be treated by surgery. So, why was Trabectedin FDA approved? Why is it any better than any other treatment for liposarcoma and leiomyosarcoma? So, this is a graph depicting Trabectedin as seen by the dotted line, and Yondelis or Trabectedin as seen by the solid line. So, the y-axis shows percent of people alive by a certain amount of months, as seen in the x-axis. So as you can see, the dotted line, Trabectedin, declines very quickly in 3 months to almost around 30%, where the Yondelis or Trabectedin line declines around to 50%. So as you can see, that Trabectedin keeps patients in a state of progression-free survival for 4.2 months as compared to Trabectedin, which keeps patients in a state of progression-free survival for 1.5 months, which is almost 3 times as longer. If we skip over to 9 months, Trabectedin treats patients around 8% of patients alive at 9 months, compared to Trabectedin, which only has around 1% of patients alive at 9 months. So Trabectedin keeps patients alive for a longer period of time. So do you have any questions so far? Yes, Aiden.
On this graph, is it overall survival or is it just progression-free survival rate?
It is patients. It's both. It's progression-free survival and patients alive. So it was conducted in its clinical trial, 518 patients, where 345 were assigned to Trabectedin and 173 were assigned to carbon. Answer your question. Jeremy?
So the graph goes all the way down to like
and 0. Does that mean like everyone who has this soft tissue disease is going to die, like it's always terminal cancer?
Yes, basically right now that's where treatments are. So like for Trabectedin, like at the around 19 months, like the last patient dies, where compared to Trabectedin, where it's around 12 or 13 months, that's where the last patient dies. Yes.
So it never touches the x-axis. Does that mean that everyone dies, or does that mean that that was when the trial, the trial was completed?
Well, it says below here, it says the number of subjects that are alive at a certain amount of months. So for 15 months for Trabectedin, that number becomes 0. For 21 months for Trabectedin, that becomes 0.
My 0 isn't actually on the x-axis.
Oh, okay.
Any other questions? All right, and I'm going to hand it over to Nick, who's going to talk about the structure
Good question.
and the mechanism.
Hi. So, Trabectedin is composed of 3 rings, an A ring, a B ring, and a C ring. Our drug binds to a group of guanines on the DNA, the minor group, and it creates these DNA adducts, which is basically DNA interacting with chemicals, and it also bends the minor group toward the major group. In this diagram in GIF, you can see that our drug, these are the 3 rings, and they form this complex. So the mechanism. So in normal replication, you have your helicase, which opens up DNA, topoisomerase that separates the 2 strands. You have your DNA polymerase that adds new nucleotides. And then you have your NER, which fixes mistakes that are made in the DNA. When Trabectedin is introduced, it first distorts the structure of DNA. It secondly creates these crosslinks and double-stranded breaks of the DNA. And what it does, what these crosslinks and double-stranded breaks prevent, is the effective function of proteins such as helicase, topoisomerase, and DNA polymerase. And it lastly prevents the effective function of the NER system to repair DNA. These double-stranded breaks that don't get fixed lead to apoptosis. And the inability to replicate DNA also leads to apoptosis. So, what is NER? NER stands for nucleotide excision repair. First, it recognizes DNA and repairs the damage. Secondly, it encompasses a family of endonucleases, which include Rad13, which is the endonuclease of fission yeast, XPG, the endonuclease of humans, and FEN1. These proteins perform the same function and have similar structures. In this image, you can see that DNA and Trabectedin have interacted with each other, and Rad13 is coming in to form a trinary structure with DNA and Trabectedin. This trinary structure leads to even more DNA damage and cell death. Are there any questions? Any confusion? Now Megan will explain the data behind this mechanism.
So we're looking at the data to understand how the mechanism works. The first question that we're going to ask is, how do we know that Trabectedin contributes to cell cycle arrest? Well, scientists did a study in wild-type fission yeast cells, and what they analyzed was how untreated cells were different from the cells treated with Trabectedin. Now, the untreated cells, as you see here, resemble bees, while at a lower concentration of the drug, you see that the cell begins to elongate, and it actually just, shows a characteristic cell arrest phenotype. And while the picture is not displayed in the data actually shows that as the drug concentration increases, you have the cells becoming smaller and rounder, and they actually stop growing after a few divisions. So what does this mean? We see that with increased amounts of Trabectedin, we also have increased amounts of the phenotype for cell cycle arrest. And so this is how we link Trabectedin with cell cycle arrest. Any questions? Yes?
So, like, according to that, the drug would cause cell cycle arrest, but how does that affect healthy cells? Because there's nothing that said it only targets cancer cells instead of healthy ones.
So you're asking, how does this affect healthy cells? Because this drug is only supposed to affect cancerous cells?
Yeah, like how does it affect healthy cells? Because like its mechanism prevents replication and growth.
Okay. I'm afraid I don't actually know the answer to that, but I could do more research into it and get back to you. Thank you. So next we have to ask, how does trabectedin cause DNA damage? And now we looked at the scientists looked at different DNA damage pathways to try to find out which pathway was the one responsible for interacting with trabectedin. And what they did was they tested the sensitivity to trabectedin of different loss of function mutations in these different DNA damage pathways. And so the first one they looked at was the UV excision repair, or the UVER pathway. And it shows that even with the loss of function mutations, these cells exhibited similar sensitivities to trabectedin as the cells in the wild type when treated. Alternatively, when they looked at the homologous recombination repair, or the HRR pathway, you see that these cells with the loss of function mutations actually exhibit an increased sensitivity to trabectedin, as even at a lower concentration of trabectedin, these cells are showing greater degradation than the wild-type cells at this higher concentration. Now, lastly, they found something very interesting in the NER, or the nucleotide excision repair pathway, especially in the loss of function mutation of RAD13. You can see that even at a very high concentration of trabectedin, the RAD13 loss of function mutation cells are able to hold out for much longer than the wild type. So what does this mean? If we know that RAD13 loss of function mutation cells do not have a working RAD13, and this results in greater tolerance to trabectedin, we can inverse that and infer that with RAD13, only with RAD13 is trabectedin able to function. Questions? Okay. And now we're going to analyze the NER pathway with RAD13. So what we know is that RAD13 with trabectedin will induce DNA damage and activate a DNA damage checkpoint. So I'd like to direct your attention to these three cells. This would be a wild-type cell without trabectedin treatment, and then this would be wild-type cells with trabectedin and RAD13 loss-of-function mutations with trabectedin. As you can see, the cells here, when treated with trabectedin, are showing the characteristic elongation in cell size. Well, alternatively, the RAD13 loss-of-function mutation cells do not exhibit this elongation. What this means is that without RAD13, trabectedin is unable to cause this cell arrest phenotype. What the scientists also did was they created a model of RAD13 with a double-stranded DNA, and they found that arginine 961, which is an arginine residue, in RAD13 occupies the same position as another arginine residue, arginine 314, in FEN1. And as we know, FEN1 and RAD13 are part of the same endonuclease family, and they're also homologous, which means that they share similar structures. And if we know that arginine 314 has an importance in FEN1, then we can infer that arginine 961 will also have an importance in RAD13. Additionally, arginine 961 is the only residue that points to the minor group, and as we know, trabectedin has to bind to a group of guanines in the DNA minor group in order to bend the DNA towards the major group. Thus, we know that arginine 961 is the point at which RAD13 and trabectedin will interact, and thus form the RAD13 DNA trabectedin ternary complex, which is composed of RAD13, trabectedin, and DNA, that ultimately results in the cell cycle arrest that's caused by the DNA damage breaks. Any questions? All right. So let's go back to earlier. We had talked about how Sun Tzu said, "Do not engage your enemy if you know that he is more powerful than you." However, even Sun Tzu must have accounted for the times that this is inevitable, and you have to challenge the enemy, no matter how powerful he may seem. Thus, he imparts on you this wisdom. Do not engage an enemy more powerful than you. But if it is unavoidable, and you have to engage the enemy, then make sure you engage it on your terms and not your enemy's terms. Trabectedin, as well as all the other drugs that are currently being made, tested, and proven to help with the cancer treatments, is humanity's way of engaging the enemy. Thank you.